on to our next set of poster presenters. The first of whom is James Todd, who's an intercalated student from Newcastle, who's going to be telling us about renal tubular epithelial cell stress.
Yes. Okay. Keep down one. Keep going. Keep going. Keep going. Keep going. There you go. Yes.
Okay. So my name's James Todd, and I'm going to talk to you today. I'm going to pose the question which is in my poster, which is: do proximal tubular epithelial cells influence T cell polarization? Now, if that doesn't mean anything to you now, hopefully it'll become clear as we go through. The reason why this question is important is that it's part of better understanding the mechanisms of chronic kidney disease. So I'm going to start by introducing chronic kidney disease. I'm going to explain our hypothesis, and then show you a few of the results and explain the conclusions. So chronic kidney disease obviously encompasses a range of conditions which all lead to progressive loss of renal function. It affects millions of patients, and obviously is a condition associated with aging, so the prevalence is increasing. The commonest cause in the UK is diabetic nephropathy. So the other common causes are hypertension, glomerulonephritis, and they all lead to tubulointerstitial fibrosis. Patients whose renal function declines sufficiently can end up with end-stage renal disease, which can often require renal replacement therapy, of course, even to sustain life. Now, tubulointerstitial fibrosis is obviously a very attractive target for treatment because it is common to so many diseases and therefore affects so many patients. But the mechanisms which drive it are not completely understood. We do, however, know that there is a putative role for T cells. So, the type of T cells implicated are CD4 helper T cells. However, the T cell lineage within this is important. We know that in the general setting that these are the principal cytokines that drive T cell polarization. However, we don't know what causes it in this setting. There is an interesting paradigm in all organ fibrosis, which is the Th1, Th2 paradigm, which suggests that Th1 cells are more antifibrotic and Th2 cells are more profibrotic. And our group has recently found more evidence to suggest this. We looked at chronic kidney disease biopsies, and we showed that there were more Th2 cells in chronic kidney disease biopsies, and importantly, we showed that the number of Th2 cells correlated with disease severity. So we might wonder that there are more Th2 polarizing cytokines driving this. And if we do wonder that, the question then becomes, what is producing those cytokines? Now, proximal tubular epithelial cells are kidney epithelial cells which line the proximal tubule. And we know that they have a role in tubulointerstitial fibrosis. We know that they secrete chemokines, which actually attracts T cells. But what we wonder is whether they produce other cytokines to then control or direct polarization of T cells once it infiltrates the kidneys. So, our hypothesis was that when stressed, proximal tubular epithelial cells will secrete this cytokine profile, which will polarize the CD4 T cells towards this Th2 rather than the Th1 lineage, and that this Th2 lineage is more fibrotic. So the aims of my project were first of all to consider which cytokines I wanted to look at and which might affect T cell polarization, then validate the methods of inducing stress in my cells, which I did with immunofluorescence, and flow cytometry. Then to look at changes in the cytokines in response to this stress, both at gene expression level, which we did with PCR and showed some changes, but they were limited, and then the results I'm going to show you a part of today, which is quantifying these changes at the protein level. And we did this with multiplex cytokine assay, which is a highly sensitive, highly specific assay, and we were actually able to show the production of cytokines which hadn't shown to be produced by proximal tube epithelial cells before. We showed changes in response to stress. But of course, the big question was in relation to the Th1 and Th2 polarization. So I'm just going to show you the results for IL-4 and IL-12, IL-4 being the most important cytokine in Th2 polarization, and IL-12 for Th1. And as you can see, it's very low levels, but generally. But we can show that there is a significant 5-fold increase in IL-4 in response to hypoxic incubation. But this is, of course, mirrored exactly in IL-12. And we showed other responses to of cytokines to stress, with other cytokines at higher concentrations. But there was no pattern to suggest that these cytokines were polarizing T cells in any particular direction. The other result I wanted to quickly show you was for primary cells, and this is again showing IL-4 and IL-12, which is reflective of a pattern that was actually quite consistent, which was that actually we found that the primary cells were more responsive generally. They produced higher levels of cytokines, sufficiently different in some cases to worry how representative our cell lines were. And actually, they also didn't show any response to stress. So, we've shown that proximal tube epithelial cells do secrete this diverse array of cytokines, and particularly responsive to hypoxic incubation. But the response of particular polarizing cytokines does not appear to be suggestive of a Th1, Th2 role. Primary proximal tube epithelial cells have a different profile to cell lines, and therefore it makes interpretation of cell line results we're in. And finally, I think we have to remember, obviously this is very speculative, but we don't know at what level the cytokine response would be, and we don't know that an equal proportional increase of Th1 and Th2. We assume that might have the same response, but we don't know, and it'd certainly be very interesting to expose these some naive T cells to this conditioned media. But to draw a conclusion from this limited data, I would say that when stressed, proximal tubular epithelial cells certainly have the potential to produce an altered cytokine profile, but this appears to be appears to be responsible for the polarization of CD4 T cells towards this Th2 response. Now, what does this mean? It means that from this data, certainly more research is certainly warranted. This is very speculative. From this data, it doesn't appear suggestive. It would be nice to explore alternative hypotheses, alternative causes of polarization. And more research is needed if we are to better understand tubulointerstitial fibrosis. We need to better understand it to be able to develop therapeutics, and it's only then that sort of research into this field can start to have a meaningful impact on the millions of patients with chronic kidney disease. But unfortunately, that is still a long way off. So I want to say thank you very much to my supervisor, Professor Sheeran, and obviously thank you so much to Kidney Research UK, without which I couldn't have done this year, and I'd like to welcome any questions. Thank you very much.
Very much. Yeah, up the back.
Hi, sorry, well
Hi.
done, that was great. Two questions. My question has two parts, if that's all right. One, why did you use the condition which you used? So you used hypoxia, you also use TNF-alpha, you also use cobalt chloride.
Yeah.
And the second part of the question is, why did you see differences in the cytokine profiles between those different conditions?
Between the different stresses. TNF-alpha was used predominantly, we actually thought it would be more as a positive control to actually look for a response. It's not particularly replicative of in vivo environment. We were trying to mimic the fibrotic environment, so we were trying to stress them in a way that would be like fibrotic. So we know that hypoxia, cobalt chloride stabilizes HIF1 alpha, so though it doesn't actually induce hypoxia, it mimics hypoxia, and so cells respond in that way. So that's why we used cobalt chloride, and obviously the hypoxic incubation actually causes hypoxia, and then hydrogen peroxide induces oxidative stress, which we also know occurs in fibrosis. So that's why we used those treatments. Obviously, it's a very long way away from mimicking any sort of in vivo environment, but we thought that was the best way of doing it.
Great. In the interest of time, we're going to move on.
Thank you
very much.
Cheers.